Welcome to the second video of APRICOT. In the first video, I talked about the development of the software for the identification of RNA-binding proteins. In this video, I will be talking about different components that are involved in the algorithm to execute an analysis of query proteins for their function characterization. APRICOT can be explained by three parts: input, analysis modules, and output. Input is what user provides to the software. Analysis modules are everything inbuilt. Users can define which analysis module to run. However, by default, APRICOT runs each of its modules. And finally, output is what users receive at the end. There are two input APRICOT requires from the user. One is query protein sets. It could be a set of 10 proteins, it could be single protein, or an entire proteome can be given. And second set of information that APRICOT requires is function of interest, which can be defined by user by providing mesh term. Now, for example, as I'm interested in RNA-binding proteins, I will be providing terms such as RNA-bind, or I can go more specific by giving the domain family names, which are RRM or KH, cold shock domain, and so on. The analysis module can be in principle divided into two parts: primary analysis, which includes all the modules which are required for processing the input by the user, and then secondary analysis is what uses the information from the primary analysis. At the end, user get a data set which comes out of the analysis, where they can select the proteins of their interest and further annotate them by functional annotations such as tertiary structure, localization, and so on. So I will in later slides talk more about these modules in detail, but before we go into that, I would like to explain the data sources that we use in APRICOT. So APRICOT is majorly dependent on functional domains, and there are two data sources that we use. One is conserved domain database, or CDD, and second is InterPro. CDD is a collection of about 10 domain databases. Together forms 50,000 entries, which are the models of domains present as position-specific scoring metrics. CDD uses 3D structure to define sequence structure function relationship, and it uses RPS BLAST to query these position specific scoring metrics. RPS BLAST is another variant of BLAST, where BLAST can be used to query proteins. RPS BLAST can be used to query position specific scoring metrics. Second set of dataset is InterPro, which is also a collection of about 10 domain databases. Together forms about 30,000 entries. It is a resource for protein signature, and it uses InterPro scan for the signature recognition in the query proteins. InterPro scan combines several database specific tools such as BLAST methods, weight matrix, hidden Markov model, and so on. The reason behind the use of these two data sources are that there is only a small set of domains which are in common between these two. Also, the tools that they use, the algorithm of PSSM and hidden Markov model, are completely different from each other. PSSM is very suitable for the domains which are majorly uncapped, whereas hidden Markov model also works well with the capped motif. So now I'm going back to the pipeline, where two input sets are given by the user, one being protein sets, and another is the mesh term, which defines the function of interest for the extraction of the domains. In case the mesh term is provided by the user, APRICOT knows a certain class of domain that should be selected from the domain database, but in case if it's not given by the user, it takes all the function classes into consideration. The protein IDs are queried into UniProt knowledge base in order to extract annotations and amino acid sequence, and the term provided by the user are queried inside the domain database to select functionally relevant domain of interest. Using the tools of CDT and InterPro, which are dependent on PSSM and hidden Markov model, the amino acid sequences are characterized by domains. In this case, we do not particularly filter out any function class, but we select all the domains that might be possible in the amino acid sequence. However, in the later stage, the domain that we selected based on our interest, we use them to filter only those proteins out which do not have domain of interest and select those which have domain of interest. At the end, the information from the database and the information identified by us are combined and characterized by physicochemical properties. Using additional tools such as RaptorX, PSORT-B, PDB database, we can further annotate the proteins of interest and at the end get the result summary and visualize the data. So in the next video, I will talk about APRICOT availability. I will combine the video with different components of the software in terms of technical detail. You can leave us your question and feedback. You can find our code on PyPy. Catch you in the next video.